Well, good morning, everybody. First of all, I want to welcome those of you who are on WebEx and remind you that if you have questions, please just email them to us. And also, can I ask you to turn your phones on vibrate and your pagers as well? Appreciate that. And we'll have questions and answers at the end. So today's going to be a little bit different from our typical grand rounds. Usually we talk about diseases and the children who have them. But there are many instances where the only way we're really going to understand more about the disorders is by doing preclinical studies in animal models. And today we're going to hear from Dr Corbin, who is one of our senior investigators in the Children's Research Institute, who's been studying animal models of autism. And I think what we are understanding more and more about autism is that it's kind of a common, there's a final common pathway with the social skills deficits and the unusual behaviors, but that there can be many ways of getting there, so that children who are prematures are at greater risk for it. Those who have particular genetic disorders are at risk for it. And what Josh has done is to look at one of these disorders, Fragile X syndrome, in which there's a very high prevalence of autism, to develop an animal model and to study the limbic system there to understand more about autism. So to introduce Dr. Corbin, I'd like to introduce Dr. Vittorio Gallo, who is the director of our Center for Neuroscience Research. Dr. Gallo.
Thank you, Mark, and welcome everybody to this morning Grand Rounds. It's a real pleasure to introduce Josh Corbin. And Josh was recruited to the center 10 years ago here at the Center for Neuroscience Research, and he's also an investigator at the Intellectual and Developmental Disabilities Research Center, which I direct. Josh has developed a really exciting research program since he was recruited here 10 years ago from Georgetown University, where he was in the neuroscience department. And as Mark mentioned, he has really been able to, in this environment, which is strong both in research and in clinical programs, he has really been able to develop a very exciting program on social behavioral disorders and autism in mouse models of these disorders. He has been NIH funded with several grants for the past 10 years and is currently funded with 2 R01 grants, and has also received funding from many foundations, including the Fragile X Foundation and Autism Speaks. He serves on the scientific review panel of many of these foundations, and he represents this hospital, this institution, with those foundations as well. His work has been prominently published. He has published some very beautiful papers looking at the relationships between development of specific regions of the hypothalamus and the brain and abnormalities in behavior. His work has been published in top neuroscience journals, including Nature Neuroscience and Neuron. So it's a very great pleasure for me to introduce Josh. I want to say that he also serves on 2 important neuroscience and autism initiatives here. One is the George Washington University Autism Institute and the University of Maryland Autism Initiative. So welcome, Josh, and thanks for agreeing to give this talk, and welcome everybody.
Thank you, Mark and Vittorio, for those kind introductions. I would also like to thank Mario Salini for inviting me to this forum and present. I think this is a great opportunity to bridge the gap that we have between the clinic and research, and to let you know a little bit about what goes on in the Center for Neuroscience Research on the 6th floor. So, I want this. All right. So I'm going to tell you today is the work that's been going on in my lab for over about 10 years now in terms of understanding the biology of autism. So these are the learning objectives and the goals that I have for you to understand at the end of my talk. So the first is to identify the unknowns regarding the biology of autism. The second is related to the third, is really about how we go about in the laboratory of trying to understand the neurobiology of autism and then use that basic understanding to translate it to potential therapeutic outcomes. So many of you are probably familiar with this, statistics and some of the biology and behavioral consequences that are characteristic of autism, but I just want to quickly go over some background. So it's a highly pervasive neurodevelopmental disorder. It affects between 1 in 68, the latest CDC numbers, 1 in 100 worldwide. The incidence appears to be about globally the same. So put it at somewhere in those numbers, though those numbers are not exact. There's a reliable diagnosis by age 2, but symptoms can appear as early as 6 months of age. And this is actually critical to understanding what's going on in the brain, because the symptoms of autism can be picked up as early as 6 months. This means that what is happening in the brain developmentally is happening at very, very early stages. At the latest, events that are happening as late as 6 months, but probably most likely things that are happening that are seeded during fetal development. The financial cost is huge. The most promising current treatments are not pharmacological. They are these early behavioral interventions that if you get the kids early by age 4 or 5 and do intensive behavioral intervention, we heard this about a month ago from Sally Rogers in her talk here, and her development of the early Denver START model is one of these examples. There appears to be some very positive outcomes for children who are already on the spectrum. So combined with this, there really remains a tremendous unmet need for novel therapeutics and novel approaches to deal with the complexities that individuals face that have autism. So this is also something that everybody's probably familiar with. So the DSM-5, 2013, it was upgraded. Autism now includes these 2, not 3 characteristics. It used to include language, but language deficits are no longer part of the diagnosis for autism. So the autism diagnosis includes deficits in social communication, deficits in social reciprocity, as well as repetitive behavior and restricted interest. Those together form autism spectrum disorder. Again, language is now out of it, although individuals with autism can be dealing with language issues, but it is no longer part of the core criteria for diagnosis. Something else that's quite comorbid with autism, but I do not believe is actually part of the criteria for a diagnosis, is the stress and anxiety that individuals with autism face throughout their lifetime. This is something that my laboratory has been getting into, and I'm going to show you some of that data. But I think this is very important to keep in mind because this also can be related biologically to the social communication deficits as well as the repetitive behaviors that are observed. So my lab has been focusing on addressing this question over the past 12 years, 10 since I've joined Children's National Medical Center, is we really want to understand the basic biology of what's happening in the brain in the systems that relate to social and anxiety-like behavior, and understand this from an embryological standpoint. So our question is, what are the embryonic genetic processes governing development of brain systems that control these behaviors, which are limbic system-based, and then secondarily, with that information, how can we translate this to a deeper understanding of the biological underpinnings of autism? I'm going to tell you how we've been addressing this question using animal models and what that might mean for potential therapeutics and treatments for individuals with autism. Okay, so this is the brain region, the brain circuit that my lab is interested in. This is a drawing done by a former postdoctoral fellow lab who is not only an outstanding scientist, but an accomplished artist. So what this defines in the human brain are the structures of the limbic system. They include the hippocampus, the hypothalamus, and the amygdala. I'm primarily going to be talking about the amygdala, which is the structure that processes social and emotional information. These systems are highly conserved throughout evolution. So This is a rodent brain, obviously not the scale, but what you can see here is that these structures are basically the same. The rodent system is a little bit more olfactory-based than the primate brain, so they have a larger olfactory processing center. But in general, the structures are highly, highly conserved, including the amygdala and hypothalamus. So across species, across vertebrate species, this limbic system is dedicated primarily as a cue detector for processing information with emotional, social, or survival relevance. And this is highly important for autism because it's these structures, particularly the amygdala, that is known to be, through many, many, many studies, to be dysfunctional in autism. It's not quite processing social information properly. So in terms of understanding autism, this is a complex disorder in which there are multiple questions at numerous levels that the field of autism research is currently confronting. And this is sort of broken down simplistically. You can break these down further, but there are 4 basic domains in which questions are being asked at different levels with different approaches. So one is the unknown of molecular pathologies. What is happening at the biochemical and molecular level in the developing brains of individuals that leads to social deficits and repetitive deficits in people with autism? It makes autism, not some sort of other brain disorder, for example, like schizophrenia. Related to that are the brain developmental defects. Autism is a neurodevelopmental disorder. That means that during the trajectories of development, starting from fetal development when the nervous system is first being laid down, through later stages, postnatally when the nervous system is being wired up, something is atypical. And this is atypical typically due to both de novo and inherited genetic mutations that can reach a certain threshold, resulting in the autistic phenotype, but also probably related in some aspects that are really not clear yet to non-genetic factors such as environmental triggers or epigenetic triggers. The best characterized of these so far are paternal age. The older the father gets, the higher the risk for autism in the child, as well as maternal infection during the 2nd trimester. Those are the best well-characterized environmental triggers for autism on a genetically susceptible background. So, sort of tackling this black box of autism and addressing these questions requires a multidisciplinary approaches that happen at numerous levels, from the clinical level down to the basic research level in laboratory. And this can include work on humans, understanding functional brain imaging in individuals with typical developing brains as well as atypical developing brains, DTI for understanding functional connectivity in the brain, animal models, which my lab approaches and many other lab approaches, animal models of both brain development as well as animal models of autism, epidemiology, really understanding where and to what extent autism is happening across the globe, and to get at hotspots, if there are any, of autism, and relate this to the state of the mother during pregnancy, as well as efforts for gene sequencing, just going in and large-scale sequencing of multiple human genomes in the thousands to understand whether the genetic susceptibilities for autism. There's been a lot of progress in all of these areas over the past 5 years. There's an exponential curve in understanding, which has been really tremendous. However, there are still a number of unknowns within each area. And I want to quote Donald Rumsfeld in terms of, I think his original quote was understanding the link between the evidence or lack of evidence for Iraqi weapons of mass destruction and terrorism about 10 years ago. But whatever you think of what was going on then, and whatever you think of this individual, his quote, I think, is best applied for understanding where we are on the stage of autism. So, Donald Rumsfeld said, there are known knowns, that is, there are things that we know. We also know there are known unknowns, that is to say, we know that there are some things that we don't know. But there are also unknown unknowns, the ones that we don't know. So about 15, 20 years ago in the field of autism was the unknown unknowns. It was a complete black box. Nobody knew what was going on. You just saw the behavioral outcomes of individuals that were dealing with autism. But I think that we've now moved to the stage of where we are with the known unknowns. We know what we don't know. And this is really, really important because then we can really start tackling this at the levels that I was explaining before. So my lab is facing the known unknown of what is going on at the biological level, developmentally, that leads to an autistic phenotype. And we study in the laboratory 4 floors above us animal models. Okay, so I'm going to tell you about the approaches that we take, and in this I'm going to tell you two stories. So one is what Dr. Batshaw nicely introduced about my work, which is our translational research program, where we actually take the best models of autism that we can find, and they're out there in the field. They're not perfect, they of course have limitations, but they can get us pretty far to understanding the biology of autism, we believe. And this is what I would call is a translational research approach. But the ultimate goal of this is to contribute to perhaps what might be therapeutics for treating specific deficits in autism. My lab is specifically interested in understanding the social deficits as well as the anxiety deficits that individuals with autism are dealing with. The second approach is one that sort of, in the push of NIH for translational research, kind of gets lost. And one thing I want to get across is the importance of just basic research. Just the idea of saying, I have a question, I want to understand how this works, and then pursuing curiosity-driven research to come up with an answer. I would call this, it's been called basic research, but let's also call it fundamental research, because it's fundamental to our understanding of the biology of any system that one is interested in. So this is the second approach that my lab takes. And this is really understanding and using animal models of neurodevelopment, just really using animal models to understand the basic processes of how the limbic system is put together developmentally. And I'm going to tell you about a program that we're just beginning to develop. So it's more of an idea at this point. We think that we could leverage this information for developing unique biomarker sets for understanding individual genetic susceptibilities in individuals with autism. Okay, so ultimately, we believe that both components, a translational research program, but also at the same time a fundamental basic research program, are essential to come together to addressing the problem, the black box of autism, for developing treatments down the road. So the first program that I'm going to tell you about, the first approach that we've been taking, is this translational research approach, which we have undergone over the past 8 years or so, a program in which we've been studying one animal model of autism, which is Fragile X syndrome, and I'll tell you a little bit more about that, and our efforts to both, one, understand the problem that's happening in the brain, and two, to correct it, come up with novel therapeutics. So in talking about that story, we've turned to this animal model of Fragile X. So as I alluded to a little bit earlier, so autism is a complex genetic disorder with environmental triggers. That means one can liken it to cancer, in which for about 90% of cancer cases, there's a complex series of susceptibility genes that an individual is carrying, and there might be environmental triggers, or there might not, put somebody over that threshold to develop a tumor. But in about 10% of cases of cancer and autism, these are due to single sort of ticking time bombs in the context of cancer, but in individuals that have autism, these are syndromic disorders. If you have a single gene mutation in some of these genes, boom, you're going to get that disorder. And these just list some of these disorders that many of you, I'm sure, are very, very familiar with, much more than I am. And one of the ones that we study, and others study, is Fragile X syndrome. So Fragile X syndrome represents only about 1 to 2 cases of autism. So it's a small contribution to the autism spectrum. But of individuals that have Fragile X, about 30 to 50% fulfill all the criteria for autism. The rest are pretty close, more or less. They can be displaying significant social defects, but maybe do not reach the threshold for autism. So we and others have been using this as a model, as a simplistic model, to understand the biology of the autism. Now, the problem in Fragile X, which I'm not going to get too much into, but I just want to touch upon, really happens at the level of neuronal connectivity, at the level of synapses. Synapses within Fragile X syndrome, this is primarily work by others, do not form properly. This is very well characterized. This is a deficit that appears to be happening in autism. Something is happening during the early stages of neural development and synapse formation that's just not quite manifesting properly. So in terms of the region of the brain that my lab is interested in, I introduced it to the limbic system, the social brain. One of the key structures of the social brain is the amygdala. This is the major hub for processing information that has social and emotional relevance. And there's numerous studies, two of which are shown here, which have characterized individuals with Fragile X as having a dysfunctional amygdala at the level of fMRI imaging. So if you present somebody with fearful faces as opposed to happy faces, the amygdala is usually activated. But in individuals with Fragile X syndrome and individuals with autism, the regulation, the processing of this information within the amygdala is dysfunctional. Sometimes it's higher, sometimes it's lower, but it's definitely off the scale what would be considered neurotypical. So my lab has been focusing on, so this is now the rodent brain. I showed you some schematics of the human brain, but if you take the cross-section of the rodent brain, the amygdala sits down here. It processes social information, and this is just blowing up the amygdala at a circuit level. Suffice to say that the amygdala is under heavy GABAergic control. That is, so GABAergic interneurons are inhibitory neurons of the brain. Essentially what they do is they work with excitatory neurons to keep your brain functioning in balance, neuronal circuits not firing too much, not too little, for proper neuronal communication. If you lose this inhibition too much, or too much excitation, you hit a seizure threshold. But below that, you can have imbalances where circuits are functioning abnormally. Now, the amygdala is under heavy GABAergic control. That is kind of tamped down. You don't want an overactive amygdala because then you will be responding to everything in a heightened state of fear or heightened state of emotionality. So this GABAergic system is critically important. So using the animal model of Fragile X, is a very well-characterized animal model of Fragile X that replicates the human disorder to the best the animal can replicate a human disorder, is we began to ask two questions. And these have therapeutic outcomes in that we're first really want to know what's the problem. Until we can correct the problem, we have to know what it is. Now, that was our first question. And then the second question is, once we get a handle on the problem, we're not going to figure everything out, but just get enough information to understand, well, how can we fix it? So this translational program, as well as other work in my laboratory and work across our center, really focuses on using animal models. And just to give you sort of a little flavor of the way we approach the problem. And of course, any of you are more than welcome to come up to our labs at any time, and we're happy to give you a tour and show you more details of this. But just to give you a flavor of the way we approach the problems, so we use genetically engineered mice with mutations in autism genes, or autism-related genes, Fragile X being one of them. And then we need to see what's going on, understand what's going on, record what's going on. We use advanced microscopy, example shown here. We use electrophysiology to listen in at a single cell level and a multi-cell level to what the brain is, how it's talking to itself in terms of electrophysiological properties. We understand, we try and manipulate these circuits, using state-of-the-art tools such as optogenetics, using light to turn circuits on and off. And then our outcome is animal behavior. These projects are multidisciplinary and involve collaborations across the board, across our own hospitals as well as other institutions across the globe. So back to the Fragile X story. Okay, so here's a bit of data. So this is our animal model of Fragile X. The nomenclature for the Fragile X animal model is FMR1KO. Okay. So what we do is now we're looking in the amygdala of Fragile X animals. We take a brain slice. We can keep this slice alive for up to 24 hours or as long as the graduate student or postdoc can stay awake doing these experiments. And what we do is we can go in with this electrical recording pipette, and we can record, we can listen in with this neuronal stethoscope to single neurons. How is a single neuron communicating at the electrophysiological, at the electrical level? And brain communication happens by chemical electrical signals. So if we go into what is a wild type or a neurotypical brain, we can see that there's a certain electrical activity. Now, if we go into the Fragile X brain, what we can see is that there's a heightened state of electrical activity. These brains are hyperactive. The amygdala is hyperactive. So this means that there's an imbalance between excitation and inhibition. It's seesawed in this direction. There's either too low inhibition, too much excitation, or both, creating this imbalance. And this imbalance, or this neuronal excitation imbalances, are what is now widely recognized across the field as the neuronal correlate of what is happening in the brain across neurodevelopmental disorders, from autism to schizophrenia to a whole host of other disorders. So what we see is this imbalance, okay? Now, that's just a snippet of data that we published a handful of years ago, summarizing work that we had carried out over a number of years, published in a number of papers, up to some very recent work that we very recently published. We came up with this model in the Fragile X amygdala, that this is a synapse, this is an inhibitory neuronal terminal, this is the synaptic cleft here, this is the postsynaptic part of the neuron with all the different GABA receptors. There's a number of different types here. And what we discovered is that these interneurons are making less GABA, there's less of these connections, and their connections are effective also in the sense that there's less and incorrect receptors in here. So there's a whole problem happening at the level of the synapse, with the outcome being that there's an imbalance in neuronal communication tilting towards overexcitation. So the goal is to bring this back in balance. We don't want this imbalance because it means that you have an amygdala that is not functioning properly in our animal model of Fragile X. So with that in mind, we wanted to address the second question is, can we fix this problem? Because this is now where we step closer using this animal model, hopefully to therapeutics. So in two studies, we have been able to correct this problem using drugs that affect the GABAergic system. The first set of drugs were very dirty drugs. The second set of drugs were possibly something that could be actually used in the clinic. So again, we have this imbalance between excitation and inhibition, and this is the data that I showed you before. This hyperexcitability at the neuronal level. Too much electrical activity, and we can quantitate it here. So what we do is we take a GABA agonist called VIP or gabadoxol. It is a drug that has made it to phase 2 clinical trials as a sleep aid, but failed there due to tremendous side effects. It's a pretty dirty drug. It affects a number of different GABAergic subreceptors. But in our hands, we weren't really worried about the behavior. We just wanted to see if targeting the GABAergic system can fix the problem. And what you can see here is that when we add the drug within this assay, it brings the electrical activity down from this hyperexcitability back to normal, back to neurotypical, right? And we can quantify that. It actually doesn't overdo it or underdo it. It actually does it perfectly at the concentrations that we use. So we were quite excited about this and really did not know where to turn, about how to translate it. We're in the laboratory looking at things in slices. So we were approached, I'm going to just come back to the past slide. We were approached through our connections through the Fragile X funding that we had by a therapeutic company that's in Cambridge, Massachusetts called Sage Therapeutics, whose only goal is to develop GABAergic receptors and NMDA receptors for excitatory. So this was a few years ago, but in the meantime, what has happened is I just kind of did a Google search as I was preparing this, and it turns out that a therapeutic company named Autism Therapeutics has just, is taking gabadoxol, or THIP, solely based on our results, and using it, putting it in phase 1 or phase 2, I'm not really sure, clinical trials for individuals with autism. It's a little bit scary because this is this failed in clinical trials at phase 2. However, the clinical trials were done in neurotypical individuals, perhaps in a neuroatypical brain, because it's different. It may work, but I was a little surprised to see this, but maybe it will work. So that experiment has directly translated to this approach by Autism Therapeutics. But what we and others really wanted to do was use targeting of specific receptors that we found dysfunctional within the Fragile X brain. We didn't want to globally get them like FIP, so we were approached by Sage Therapeutics. Sage Therapeutics at that time was in the beginnings of a Fragile X program in which they were really interested in dealing with the anxiety and social deficits that were present in individuals with Fragile X. And they had a hypothesis that fit with our data, this GABA-A receptor hypothesis, that by targeting GABA receptors within the amygdala, they can really tamp down or in part ameliorate the deficits in both anxiety and social communication that exist within Fragile X, and if that works, use that as a platform to jump up to the more heterogeneous cases of autism. So using the same assay, we took their drugs, and this is just showing the data in a little bit of a different way, and what we found is that we have so these are their compounds that we had tested in our lab a few years ago and recently published, in which these are the control brains and these are the Fragile X brains. So what you can see here, just focusing on this data, is that if you give a certain concentration of the drug within that assay that I was showing you, put it in the bath with the brain slice and record electrical activity, what you can see is that there's a boost in inhibition. And this boost in inhibition in the mutant amygdalas is not significant from the normal level of inhibition that one sees in the presence of a control drug or their drug. So this and this is the same, meaning that we've boosted this up essentially back to normal levels, which is the goal. Essentially what we've done with FIP, we've now a much more targeted drug, one of which they has passed or is in phase 3 clinical trials for intractable seizures. So with that in mind, So that's just sort of an overview. I'm happy to provide anybody more details, and I can also point you to publications, but this is just an overview of how we've taken that information and we're moving that information hopefully to the clinic. So we've identified deficits, we've defined amygdala dysfunction at the GABAergic level. By targeting a subtype of GABAergic transmission with a drug, we're able to correct it, and now we've taken it further by teaming up with Sage Therapeutics to test the novel compounds that are very specific to the GABA receptors that we believe are dysfunctional. Okay, so the second part of the story that I briefly want to tell you about is this concept of fundamental research, really just exploring the basic biology of how the limbic system, how the amygdala is put together developmentally, and then how that information can then be leveraged to begin to understand autism. And in our case, we believe that what we're going to be touching upon is biomarkers that may be used for precision medicine for understanding unique genetic deficits, a unique genetic load that individuals with autism are carrying. So I'm going to tell you a little bit about our recent data in terms of understanding the genetic mechanisms for development of the social brain and how that information, we believe, can translate for understanding biomarkers. So work in my lab, the sort of other side of my lab, the basic research side, from the translational research side, has been addressing these basic questions of how does the limbic system develop. So we have published a number of papers understanding and unraveling unique genetic and developmental events, fetal developmental events, that lay down, that are required for putting together the amygdala. Events that are unique and different for putting together other brain structures, which is actually important when you think about autism, because autism is not a global brain deficit. It's a deficit in certain regions of the brain that control processing social information, as well as processing information that controls repetitive behavior, such as the motor area. Then when we think about specific brain regions that are atypically developing, we have to understand how those brain regions develop. There's not one single game plan for the entire brain. Each structure seems to have, in part, unique rules of how they develop. So what we determined over the years was unique genetic and developmental rules for putting together neurons of the limbic system. This is work that's published a handful of years ago. And just to orient you, this is the developing mouse brain, and these are neurons, developing neurons that are being born in the fetal brain. They're going to become solely part of the limbic system and solely part of the amygdala. So again, we try and keep our eyes on the bigger prize, because sometimes we can get distracted by looking at the smaller picture, which is the amygdala. But now thinking about this in the context of autism, what we discovered most recently in a paper that was published in Neuron, and again, the artwork of my former postdoc got us to cover, as well as there's a YouTube video on this. So what we learned is that there's a specific gene that sets up the stress system of the brain within the limbic system. This is a fetal gene that is only expressed during fetal development, yet it sets in motion a cascade of events that makes the stress system of the brain. Much later behavioral events are seeded by these embryonic programs that are happening during fetal development. And again, I'm happy to discuss any of these aspects in more detail if folks are interested. Okay, so what we did is we created a genetic deletion of this gene. We removed it from the genome, and we let the animals survive. So I'm going to show you a mouse in a cage. So this is a control mouse, a neurotypical developing mouse. This is a rat odor bedding. Okay, this is a normal, very, very stressful cue to an animal. They smell this. This is a predator odor. They freak out, and you can see this animal essentially freaking out. They're climbing, they're rearing, they're trying to escape. They want to get away from what they perceive as a predator threat, right? Now, these are our stressless mice. By removing this single embryonic gene, we created stressless mice. These mice, they'll check it out. They're curious. We didn't remove their curiosity, but what we removed is their ability to respond to a stressful cue. So they're interested, but essentially they don't freak out. And this is all quantifiable. So what we found, so the stress response in animals and humans across species is a cortisol or corticosterone peripheral response that can be measured in blood that is triggered from the brain. So what we found, so if you take a control animal that I showed you and you put it in this rat odor as opposed to benign odor, their HPA axis is through the roof. Their corticosterol levels are through the roof, right? But if you look at our mutant mice, basically, physiologically, they're not responding to a stressor. Behaviorally not responding to a stressor, physiologically not responding to a stressor. Okay, so coming back to the central focus in which we try and look at our studies, sometimes we move away from this autism, but we always just sort of bring ourselves back. What does this have to do with autism, right? A mouse running around a cage, basically afraid of rat poop or not afraid of rat poop. What does this have to do with autism? Well, again, coming back to the core deficits that are associated with autism, social communication, repetitive behavior, and the stress and anxiety, which probably underlies both and I think is undercharacterized in the field. So there's a lot of recent literature that the cortisol response, the stress response with children with autism is dysfunctional. So perhaps we've touched upon the genetic makeup, the genetic blueprints for setting up the stress system. It may have something to do with autism. So behavioral and physiology can be linked to autism. And again, I bring this up, I'm not saying this definitively. These are questions. We're doing research, exploratory research. We don't know how far we can take this, but we're going to take it as far as we think that we can go with it and how much it actually does relate to autism. The second thing is genes. So I mentioned our gene of interest. I introduced you to our gene of interest, which is this gene, this embryonic developmental gene called DBX1. It's a transcription factor, which means it's high in the pyramid of regulating other genes. It turns genes on, turns genes off, which sets in motion a cascade of events to make neurons that are going to become part of the amygdala during fetal development. Now, we have, in collaboration with Kazui Hashimoto-Tori in our department, we have, in the process of trying to understand what are the gene cascades that are regulated by DBX1 in establishing and setting up the limbic system. One of our hypotheses going into this is that we would come across genes that were known and recognized autism susceptibility genes. DBX1 itself is not an autism susceptibility gene because it's just so important in many developmental events, including making the breathing neurons of the hindbrain. So if an individual has a mutation while they're developing in DBX1, autism is probably the least of their worries. They're not going to develop the breathing centers. But the cascades that DBX1 is controlling to make neurons specifically of the amygdala, we believe that we are beginning to uncover, and about 10% of these genes are validated autism susceptibility genes. So when we came up with these findings, which we're still in the process of looking more deeply, we kind of had the aha moment of, wow, we think we might be hitting on gene networks that are related to limbic system dysfunction with an autism. So the point that I want to get across of this, and I'm going to tell you how we might be moving forward with it, is that by studying basic mechanisms, these fundamental understanding of how these systems that are dysfunctional autism are put together during fetal development, by studying these basic mechanisms of the social brain, we believe that we've identified an amygdala-based autism susceptibility gene set. So with Lauren Kenworthy, we're in the very beginnings of discussion of, well, how can we actually translate this? What does this mean, right? Can this be something where we can actually design precision medicine biomarkers? So we'd like to use this knowledge, generate a unique biomarker set to screen for mutations in the social brain gene set, meaning that at a precision medicine level, each individual with autism is most likely carrying a series of mutations that led to their autism. These are different from individual to individual, most likely. Now, these susceptibility genes, which hit a certain threshold, some of them are genes that are likely involved in development and function of the limbic system, development of the amygdala. So if we can come up with a panel of genes that an individual can be screened by using, from our basic knowledge, a chip or do it by sequencing, that we might be able to develop some sort of biomarker set that could be designed for individuals to understand what genetic load is that individual carrying, and to use that for therapeutic interventions that would be individual-based. So what we're working on in the laboratory is this understanding the social gene brain set in rodents. These genes are, when I tell you that they're autism susceptibility genes, these are human autism susceptibility genes. The genes are highly conserved across species. We are in the beginning of teaming up with Lauren Kenworthy's team at the Center for Autism Spectrum Disorders and just start taking cheek swabs of individuals and their parents with autism so that we can understand the genetic load that the parents are carrying and what that genetic susceptibility is in the individuals. we're looking for partners, perhaps at CHOP, perhaps at other places, very beginning of this project, in which we can then sequence their genes or use a SNP chip to really understand what genetic load are they carrying, are these individuals carrying in their autism susceptibility gene set, and then perhaps combine this with Kevin Pelfrey, who is just being recruited to George Washington and will be heading up their autism initiative, perhaps to image these individuals related to what's actually going on in the brain, as well as behavioral assessment to really put together genotype, phenotype, and brain dysfunction in individuals. So that's how we believe that this information can be used to translate for biomarker discovery. Okay, so in summary, what I showed you is two approaches that my laboratory is taking for, specifically in the context of animal models, for cracking the black box of autism. So the first approach was what would be considered a more translational research approach, in which we're using an animal model of Fragile X syndrome to really understand how we can get to therapeutics that might be beneficial in Fragile X and beyond. And the second approach of just using basic curiosity-driven research to understand the biology of neurodevelopment of the limbic system as a potential way to develop a unique biomarker set, with the ultimate goal, of course, of bringing this together for treatment of individuals with autism. So back to the three learning tools, the three learning objectives. So hopefully in the beginning, I showed you what we don't know about autism in a broad sense. There's a lot of unknowns, but in each realm of the unknown, there's a lot becoming understood, and then our contribution to this bigger picture. And hopefully I've convinced you about how animal models can be utilized as a step in this process of developing novel therapeutics, and then how both basic and translational research is needed to be leveraged to understand and treat autism. So with that, I just want to thank the individuals that carried out this work in my laboratory over the years, a number of really, really wonderful folks who have gone on to different positions, have gone on to faculty positions, as well as individuals that are still in my lab. So the Fragile X project, with a number of individuals over the years shown here. This work was funded by Autism Speaks and FRAXA, as well as a generous donation by Larry Eichel, tribute to our Neurodevelopmental Disorders Award, made this research possible. And the work on the social brain, which is ongoing in the lab. A number of folks, many of whom are still in the lab, discovering new and wonderful things on a daily basis, as well as internal and external collaborators, and this work has been funded consistently by the NIH. And I also want to have to thank Children's National and CRI and the Center for Neuroscience Research for over the years supporting me, supporting my laboratory, supporting the directions that we take, giving great insight, financial support, all sorts of support that would not be possible without it, specifically Mark Batchel, Mendel Puckman, and Victoria Gallo. And I want to thank you for your attention, and I'm happy to take any questions.
A lot of what you're describing is also seen in children with Tourette syndrome, and I'm wondering whether anybody has looked at the amygdala or the areas that you've looked at to see whether these children who also have social problems as well as anxiety and some of the other things you've described, and even some of it seems a little bit autistic.
Right. So I'm not sure what imaging has been done in the limbic system in Tourette syndrome. I would imagine that there has been some done in the amygdala. And if they have social deficits, my guess is that it would be dysfunctional. Most of the work in the field has been focused on the motor areas, but I wouldn't be surprised if there's been some that has addressed the limbic system deficits, but I don't know specifically. But one thing that's actually been very interesting is, so over the past 5 years or so, as gene sequencing has become much more accessible and much cheaper, there have been thousands of individuals sequenced with a number of different disorders: autism, schizophrenia, Tourette syndrome. And while each disorder is unique, what has been found is that a certain percentage of genes are common to all disorders, or common across disorders. So there are definite biological commonalities that define these different disorders, as well as major differences, of course. But there are commonalities, and you guys probably know this better than I do. I think sometimes it becomes difficult to determine who has what, right? When because the determinations of a disorder are solely based on behavior. There's yet to be biomarkers that can define disorder A from disorder B, right? And the field is moving, has to move in that direction, so it can be, these are biological disorders. They need to be defined at the biological level, right? So this is emerging. The commonalities between disorders is really very, very important.
Thank you.
So, Josh, that was really beautiful. I wonder, in terms of your genetic analysis, you're taking a mutational approach, and that does make some sense. But I wonder if in parallel you might think about taking more of a modifier gene approach, because it could be genetic variants that are functioning in an epistatic sort of way. You mentioned SNP chips. So are you taking kind of a targeted SNP-based analysis to look at areas of the genome that might be linked to susceptibility and the interactions among them?
We want to go there. That's where we want to go. So the data that I showed you of the gene discovery is 3 months old, right? This is not published. This is just in the works. So we're in the process of validating those genes in the animal. And then that program that I showed you is where we hope to go over the next 5 years.
Right, because you may not find mutations, but you may find variants.
Exactly, exactly. So we want to partner up with a genetics person, you know, CHOP or Yale, wherever we can come up with somebody, of the people who sequence the thousands of genomes at a huge scale and really know how to interpret this data. So we need these different pods in which we can take our individual expertise and bring them together, but that's exactly the direction that we want to go.
Thank you.
Josh, I have a really translational question that I'd like your thoughts on, but first I just wanted to congratulate you on a really extraordinary presentation. I think it's widely acknowledged that the neuroscientists at Children's National are the smartest people here, those in Dr. Gallo's group, and yet you made this very intelligible for all of us, so congratulations. My question is this: if you have a model like, or if you have a disease like Fragile X and you have a drug like gabadoxol that's going to normalize the GABAergic transmission at the synapses he so elegantly showed, and it's really a developmental question, and let's say that drug had a acceptable safety profile, and you gave it to, you know, young kids, medium-aged kids, teenagers, adults, in whom the phenotypic expression of the disease is at various phases of development. What's going to happen to those children and young adults if you began to normalize that? Any thoughts?
So there's two really good questions in that one question, and so the first component of that is the developing brain is different at different stages. So the experiments that I showed you were done in animals that are equivalent to about late adolescence, early adulthood, right? When the systems have more or less, in a typical developing brain, been wired up. As we know, brain development in humans is basically happening until the age of 25 in males, right? You don't have a mature brain until you're within your, you know, past your first quartile of life, right? So we looked at that age in which things are pretty much laid down already, more or less, right? And we have this outcome that was pretty satisfying, right? Following up that study, we actually looked in a developing brain over time. Are these deficits that we're observing at 3 or 4 weeks of age in the mouse the same deficits that we observe at birth or in between, right? Because individuals with autism or anything, right, are different at different stages, and the brains are different at different stages. So what we actually found is that at earlier stages, we have too much GABA. You have too much inhibition, right, which is the opposite of what we saw at this stage. So these pharmacological approaches might actually have a negative outcome if they're done at the wrong time. So timing really matters, right? So the brain is dynamic, and there's also compensatory mechanisms that are trying to fix the problems that exist, right? Those also play into it. They're not the genetic lesion itself, but the response to the genetic lesion. So all this has to be taken into consideration, right, when clinical trials or the drugs are administered. So the biology can hopefully help inform that, right? But it's going to be different at different stages. The brain is going to be completely different. So there has to be readouts in individuals. What is happening in the brain at this time? Whether it's going to be through some sort of biomarker, whether it's going to be through some sort of imaging, MEG or PET or whatever, to say their GABA is up now, their GABA is down, or whatever it might be. So this is where the idea of personalized, individualized medicine has to come in. How do we treat that individual? Because everybody's going to be different, even if they're carrying the same genetic lesion. Fragile X is Fragile X, the same genetic mutation from individual to individual. But there's huge variability across time and across individuals. So it all has to be taken into consideration. And some of our other data that I didn't show you points to the fact that things are highly dynamic and different. Does that get it? Yeah.
Hi, this is Billy Short. Excellent, excellent talk. I wonder, are you involved at all in the fetal center here? Because Kathy Limperopoulos, who's one of the fetal researchers, has found in work before she came to Children's that some of the fetal studies, babies that had cerebellar hemorrhages developed autism, a high percentage of those kids. Right. And we're obviously doing a lot of MRI imaging, not only fetal, but then post-fetal in our premature population, which we know has a high incidence of autism developing later. And you may be able to partner and get some biomarkers that are MRI imaging early on some of these kids.
No, we're not, and I didn't know about that, but that's very important. And the cerebellum, I mean, it's focused on the amygdala and the limbic system, but the cerebellum is one of these other hotspots that is dysfunctional autism. So I guess in those cases, it's just a cerebellar lesion that is leading to autism, or is it other?
Yeah, that was primarily a cerebellar lesion, but I don't know how. I would have to have her come here and talk about
Because the cerebellum is, many of the genes that are involved in cerebellar development are autism susceptibility genes, and it is one of the regions of the brain that's dysfunctional autism, but nobody knows. The limbic system is easy because we know it controls social behavior, but the cerebellum, movement, cognition, it's really unknown about how that plays into the phenotypes of autism. Maybe the repetitive, but it's really a black box. I mean, bigger than, so it would be interesting if you have these specific lesions that result in autism. And whether that autism, what is that autism? How different is it from perhaps the other kinds that are not due to genetic lesions like that or insults like that?
I might tell her to come talk to you.
Sure, yeah, thanks.
Wonderful presentation. Thank you so much. My question has to do with, since autism is such a wide spectrum disorder, you have the very high functioning kids with very high IQ sometimes, and then you have the very low functioning kids who are very impaired. Is the GABAergic transmission, does it kind of go with how impaired it is based on phenotypic presentation?
Nobody knows. So again, there's no biomarkers that segregate individuals with autism. It's solely based on behavior, right? So major thinking in the field is there's autism, right? And it's somebody who's impaired language, mental retardation, no social communication is a different individual. I mean, at behavioral level, obviously, but at the genetic level, than the individual who is Asperger's, right? High functioning. Now Asperger's is lumped in with, it's not a separate disorder. It's lumped in with autism, right? They're clearly very different. But what is the biology that's different about them? Exactly what you're saying. Are their levels of neurotransmitter different, right? What is the genetic load they're carrying? Is one set that's socially dysfunctional, but okay with language, carrying a certain genetic susceptibility that's involved in social brain formation versus the genes that are involved in language formation? Unknown. I mean, this is where the field has to go.
So, hello, hi. Hi. So, considering the time point that DBX is on in the mouse, very early in fetal development, my suspicion would be that it would also affect neuronal connectivity. And I'm just wondering if you've thought about crossing these mice with like rainbow mice to see where neurons are, where they're connected to, and how that might later play a role in signaling. As opposed to, sort of the downstream elements of how many neurons there are, how many receptors there are, and how much GABA is being released in the synapses.
Yeah, so very, very insightful question. If you want to work in my lab, you're more welcome. That's on the basic side. Those are the exact approaches that we're taking. We're really trying to understand, we can tag these neurons, the descendants of these neurons using pre-lock systems, right, and reporters. And we're using those approaches to understand their destiny and their connectivity, and then see if that somehow is related to the connectivity and disconnectivity that's in autism. So we can take it farther than the genes, basically. So exactly, yeah, that's a really good question. It's exactly the types of approaches that we're doing, yeah.
Josh, that was great. I think the folks might be interested in your describing what the behaviors of these mice look like in addition to the stress behaviors. In other words, how valid are these mouse models for human disease?
Right, so I'll tell you about both of the models. The translational model for Fragile X syndrome is an incredibly valid model in terms of behavior as well for Fragile X syndrome. So as best you can replicate, and Zena Casado, sitting here, has done some work on this as well. There are a number of, so mice are social, so you can actually replicate a number of social dysfunctions in mice and measure those. So the Fragile X model work, not by my group, but by others' group, has shown that they're socially dysfunctional in a way that it mimics autism social dysfunctions. The Fragile X model has that validity, so yes. The animal model that I was showing you of DBX, or removed DBX, when it comes to the social behaviors, we have not explored that much in depth. So we don't know. We do know that when it comes to stress, these mice are not autistic because they're actually less stressed, right? They're not heightened stressed, they're less stressed. We think right now this is a better model, not for the behavior, but for the gene networks, right? And we hope to use it for that approach and then bring it back to other animal models that are more validated at the behavioral level and see how those genes overlap, right? So a kind of yes and no answer.
Well, thank you very much. It was a great presentation.
Thank you.